Then there is another interesting. Mitochondria is important from the point of view of both prelims and mains. Lot of question have been asked from mitochondria. Lot of question. Basic question was semi-autonomous organelle, and then the emerging question like 3-parent IVF, and the another important area is your DNA barcoding. DNA barcoding. This has been in lot of news. DNA barcoding has been in lot of news. DNA barcoding. DNA barcoding. We have two terms over here, DNA barcoding and universal primer technology, UPT. DNA barcoding and second is universal primer technology. Universal primer technology. Both function is same but name are different. Basics technology is also same. One is DNA barcoding and second is universal primer technology, UPT. Universal primer technology, UPT. This UPT, universal primer technology, was developed in India at CCMB Hyderabad, Center for Cellular and Molecular Biology, Hyderabad. So this is a DNA based identification process. DNA based identification process. This is based on the concept that a small segment of the genome can serve as a molecular signature or molecular marker. Based on small genome, you can try to identify the organism. This technique was developed by Dr. Lalji Singh and Dr. S.K. Verma. Lalji Singh is also the founder of an important organization called CDFD, Center for DNA Fingerprinting and Diagnostics. So Lalji Singh also pioneered the DNA fingerprinting process in India. Now, the UPT, Universal Primer Technology, is based on cytochrome B gene. This is based on cytochrome B gene found in the mitochondria. So the universal primer technology uses what? Cytochrome B gene that is found in the mitochondria. So this can be used to identify different species of animals, whether it is birds, fish or animal species from the given biological sample. That is why it can be used to detect adulteration of animal meat. For example, mutton or goat meat. Goat meat is costly, so beef is mixed as an adulterant. Beef or dog meat can be mixed into goat meat. So by DNA barcoding or universal primer technology, we can identify the adulteration that has been done in case of meat product. We can identify the adulteration that has been done in case of meat product. Another is your wildlife crime. Say a tiger skin is found. We have to first identify the color may be similar to tiger skin. It might have been painted, but it may not be original. So to identify the sample, you use this technology UPT. So it can identify adulteration of animal meat, then identify species. So this is used to curb illegal trade, in wildlife. So you get the sample, You establish what sample is there and then you prosecute them. So wildlife trade is over $19 billion annually. Large amount of wildlife product go into China. China is one of the major culprit. Different type of animal things are consumed in China for different purpose, aphrodisiac, for treatment, etc., etc. Many of these are basically cultural thoughts. It can be used for identification of invasive species. Invasive species are species coming from outside. Outside species. It can be plant, it can be animal, it can be microbe. Plant or animal. So outside species can come and can affect. This can also be used for identification of agricultural pest or biological control agent. Biological control means use of one biological organism against the other. Use of one biological organism against the other. For identification of indicator and keystone species. Biodiversity monitoring. How many varieties are there? Two species can appear to be very similar, but they can be genetically different. So that is your biodiversity monitoring. Biodiversity monitoring. Two species can appear to be very similar, but they can be distinct species. So one is your UPT and second is your barcoding. So UPT was developed in India by Lalji Singh, whereas DNA barcoding was developed by Professor D.N. Herbert in Canada. This became more popular because the name is appealing, DNA barcoding. And this is also based on identification of the unique target gene COX-1 of mitochondria. UPT uses one. UPT is based on cytochrome. We had discussed that cytochrome B gene. UPT is based on sequencing the cytochrome B gene. So you find out the sequence of the 4-letter ATGC. So UPT is on cytochrome B gene. Whereas DNA barcoding is based on what? Here the target gene is COX-1. This is the target gene. So you find out the sequence of nucleotide in the COX gene. So both UPT and barcoding is based on identification of the sequence in the sequence of nucleotide in the target gene. Now take a look over here. Universal Primer Technology. This is from Hyderabad, CCMB, Center for Cellular and Molecular Biology. So what are the application? Wildlife, detect adulteration in meat tissue, monitor distribution and migration of species. You can take out very small amount of the sample for both DNA barcoding and UPT and use it. Accurate and quick identification of species from tiny biological sample. Management of wildlife, man-animal conflict. You find out which animal is entering into the farm, exact identification, migration of species. So these are some of the other applications. Invasive species identification, agriculture pest. Recently you have locust. There can be different variety of locust. Find out the species of commercial value. Identification of biomedically important species. Ecologically important indicator and keystone species. identification of the endangered species, identifying the biological control agents, quarantine inspection. When agriculture product and other product come from other country, we do inspection to find out whether any harmful invasive pest or organism is also coming or not. So for that we use quarantine inspection, environment assessment, biodiversity monitoring. These are some of the application of DNA, barcoding and UPT. The next important